Hi, everyone. Thank you for joining this online seminar. My name is Qiu-Xing Jiang. I'm the new director for the Cryo-EM Center at the Hauptman-Woodward Medical Research Institute, which is in the downtown area right next to Roswell Park Comprehensive Cancer Center. So today I will give a presentation on Cryo-EM with a simple introduction, and then a couple of areas that Cryo-EM can be used for addressing important biomedical and biological questions. So the center set up at Hauptman-Woodward is purposed for therapeutics development, and it's going to be dedicated for both commercial and academic users. So my besides the Cryo-EM Center, I'm also running a lab of molecular physiology and biophysics. My main science programs are in three directions. The first is in membrane biology and biophysics. We are leading two directions: lipid-dependent gating of voltage-gated potassium channels, and also the ion channels in the regulated secretory pathway. The second main direction in my laboratory is the ion binding proteins, mainly the biophysics studies, especially prepare them for Cryo-EM studies. The third direction is the chemical functionalization. I will actually talk one example from each direction, so I will point them out to you. So today my talk will be oriented for a general audience. So if you want to ask a question, please stop me in the middle. I will first give a brief introduction. Then I will talk about the use of Cryo-EM for therapeutics development. And then the last three parts, I will talk about these three typical projects from my science program. First, I will give you a brief introduction about cryo-electron microscopy or cryo-EM. So cryo-EM has been a technology for structural biology since 1960s, actually. The principle for three-dimensional reconstruction of electron microscopy images was proposed quite long ago. And then during the past, almost 60 years, the technologies advanced such that the single particle cryo-EM has been able to deliver near atomic or atomic resolution structures. Because of this major advancement, three pioneers in the field was awarded Nobel Prize in 2017. So here are the images of three pioneers: Jacques Dubochet, Joachim Frank, and Richard Henderson. Dubochet developed the first method for flash freezing of the cryo-EM specimens, which is very important for preserving atomic resolution information of biological specimens. And the same method was actually also used for data collection by X-ray diffraction from frozen crystals. Joachim Frank was the person who actually pioneered the idea for using computer analysis to combine images of particles or molecules oriented in different orientation. And Richard Henderson was the one for the first time showed in 1990 that it was possible to obtain 3D reconstruction at near atomic resolution. It was the landmark paper in 1990 in Journal of Molecular Biology of the 3.0 angstrom structure of the bacterial rhodopsin. So with the recent development, currently there are four main areas of using cryo-electron microscopy. really depends on the type of specimens. If you have 2D crystals or actually tubular crystals or even small nanometer size crystals, you can perform electron crystallography. The second part is the major direction currently ongoing, which is called single particle reconstruction, which is to use computational algorithms to combine images of single particles in different orientations, such that you can actually use the images from two-dimensional space into a three-dimensional structure. The third direction is electron tomography, which is to build three-dimensional structures of ultra-thin sections that are about 100 to 200 nanometer in thickness, mainly subcellular sections or thin cells like a lot of the bacterial cells that are very thin can be flash frozen without damaging the structural information. The last area is called correlative light and electron microscopy, which is the method to bridge the resolution gap between light microscopy, which is about 100 to 200 nanometer, to electron microscopy, which is better than 10 angstroms. So there is a gap between 100 nanometer to 10 angstrom, which is 1 nanometer. So using this method, you can actually make correlation between light microscopic images and electron microscopic images. So this is the general idea. So with the recent development, especially in instrument, algorithm, detectors, and sample preparation method, it's now possible to reach pretty good resolutions for most of the specimens, single particle, electron crystallography, and micro-ED, micro-electron diffraction, which are from nanometer size to three-dimensional crystals. For all these three types of specimens, it's now possible to deliver near atomic resolution structures. The sample that we still cannot reach high resolution is the one from thin sections or ultra thin sections of tissues or cells, which is the second one. For this type, without averaging, the resolution limit is in the range of 1 to 4 nanometer. With what we call subtomogramic averaging, it's actually now possible to go better than 10 angstrom in resolution. The best has been achieved with around the 3 angstroms. So in general, to start a Cryo-EM project, especially for those in the audience who's not familiar with it, you have an EM grid, which is shown on the left side. And EM grid is 3.09 millimeters in diameter with multiple empty squares. And then these empty squares are covered with a carbon film. Shown here on the right side is a quantifoil carbon film, which contains holes in arrays. This is called a holy carbon film. And these holes are about 1 to 2 microns in diameter for single particle specimens. And our molecules are in the range about 20 angstroms to hundreds of angstroms in diameter, much smaller than the holes. And the idea is, to make a sample, the idea is to load the sample in suspension onto the grid, and then load the grids into a device which is the one, a modernized version of the device first implemented by Jacques Dubochet, which is used for flash freezing, which can freeze the block most of the specimens away and then freeze the specimen to liquid ether temperature within a couple of milliseconds. And this is called flash freezing. And the temperature change is so fast such that the small amount of specimen which was left after blotting is quickly switched from around room temperature to liquid ethane temperature or liquid nitrogen temperature, such that the water turn into glass state ice, which is non-crystalline, such that there will be no ice crystals around the molecules, and the molecular structure will not be damaged. Shown here on the right side is the thin ice across a single hole in the Holy film. I showed in the last slide. You can actually see that the individual molecules are kind of still float around in the thin ice in different locations, and this is going to allow which will allow the preservation of the specimens in the liquid nitrogen temperature, and the atomic resolution information of the protein is retained. And in these slides, I'm showing you two versions of the Cryo-EM. So Cryo-EM is different from the regular transmission electron microscopes by keeping the specimen, which we already flash frozen and maintained at liquid nitrogen temperature, and we can then transfer into the microscope, and then the microscope can take the images from the specimen while the specimen is maintained at near liquid nitrogen temperature, such that we can collect the images from the particles in suspension. As I mentioned, because of the retaining of the new atomic resolution information of the specimen in the micrograph, the micrographs contain the high resolution information. This can then be used for computational analysis. On the left side is the old version microscope, and then you can actually see this is in the middle. This is the sample pot where we can transfer the specimen into the microscope while maintaining it at liquid nitrogen temperature. On the right side is a very modern microscope, and it's a black box. Basically, the sample is inserted inside into an automatic loading system, and then being kept at liquid nitrogen temperature. And the modern microscope is much more stable and have a lot of other advantages, optical properties, so that the data collection is automatic, much faster, and can continue for an extended period of time. This will make the data collection much faster, and it's easier to collect enough data to deliver atomic resolution structure. Shown here in the next slide, I will quickly go through a few examples showing that Cryo-EM can really do a wonderful job. So on this slide, I'm showing a famous example also for structural biology, hemoglobin, a 64 kilodalton protein. It's actually possible to collect the Cryo-EM image and then use computational analysis to evaluate the quality of the image, get class averages, and then use the individual images to build a three-dimensional model. Shown here is the gray is the electron density map, and the yellow and the blue sticks are the atomic model for the molecules. Actually, using this procedure, you can actually find out three different conformations of the molecules. The R1 are the relaxed conformation, and then the tight conformation. This is actually the first example for the allosteric conformational change of a macromolecular complex. So this is actually a large complex, ribosome, 70S ribosome at 2.8 angstrom. I'm not going to go through the details. And this is one of the highest resolution structure obtained by Cryo-EM, the 334 kilodalton GDH, which is from bacteria, purified from bacteria, and using Cryo-EM, it's possible to reach 1.8 angstrom. In the bottom, as you can actually see, the electron density map really match with the atomic model, and the expected hole in the middle of the benzene ring is actually clearly visible in this side chain of a tyrosine residue. Even at resolution in the range about 2 to 10 angstroms, or even worse than near atomic resolution, you still can use Cryo-EM to answer biological questions. So here is an example. The holoenzyme of the human telomerase resolved at 8 angstrom. At this resolution, it was actually possible to identify for the first time the 10 major components of the holoenzyme, which has not been possible for decades, actually. So this is the brief introduction. If you have any other questions, you can ask me, or you can also email me later after the lecture. In the next, I will show you 4 different directions. The first is the structure-based drug design. The general idea is, we can, for X-ray crystals, this has been done for a long time. We can integrate the Cryo-EM structures at a near atomic or atomic resolution resolutions into the pipeline so that we can use the structure to optimize chemical compounds for biological complexes. This has been actually proposed a few years back. We actually started this when I first relocated my lab to University of Florida in 2015. So we picked up a sample. Sorry, let me actually bring up something actually more relevant to the current COVID-19 pandemic. So this has been a focus for many structural biologists in the past few months, or actually since SARS period in 2003 as well. Shown here on the top is the genome organization of SARS-CoV-2, and there are in total 29 proteins. The 3 main examples that are being used for druggable targets are the NSP5, non-structural protein 5, which is the main proteases important for activating the virus before its cell entry. NSP12 is the polymerase, which is important for the viral replication. S is the spike protein, which is the one important for the interaction of the virus with the cellular receptor. NSP5 has been resolved by X-ray crystallography shown here on this side. And today there is a newly released structure of the NSP5 in complex with different compounds that are coming out of the lab in China. And one of the compounds appears to be highly potent and showing very high promise for clinical applications. In the middle is the Cryo-EM structure of the NSP12 in complex with NSP7 and NSP8. This is actually the the molecular model. And this is actually one of the main relatively conserved proteins among a significant number of coronaviruses, and also one of the main targets for drug development. On the right side is the spike protein and its receptor called ACE2, and the ACE2 form a dimer and combines to a transmembrane protein. And you can think of this as the surface of the virus. It has the spike protein which protruding out, interact with the receptor on the surface of the cell membrane. This is the membrane of the cell, and ACE2 is actually presented on the cell surface. This interaction, then there is a protease, NSP5 or another protease. Actually, mainly the proteases in the extracellular side of the human cell, which will come and clip the spike protein, release the S2 part of the spike protein, which then mediates the fusion of the cell membrane with the viral membrane. And this is the required event for the cell entry. So our selected target is PRMT5, which is a methyltransferase with significantly higher activity in a broad spectrum of cancer, of the cancer cells. Because of that, we choose this as a target. And our collaborators already have multiple compounds in the cell-based, in the in vitro assay. Two of the compounds, 11-2F and 11-9F, were able to reach submicromolar affinity. However, in the cell-based assay, we found that there is a problem for bioavailability issue because all these compounds now have IC50 above micromolar affinity. So our main question is how to redesign them so that we can actually enhance the efficacy in the cancer cells. This is the cancer cell survival assay. So to do that, we need the structures. So we did two structures. Shown here is the first one, which is the enzyme in the apple state. Each enzyme, each molecule contains 8 subunits, 4 of the PRMT5 and 4 of the MAP50, which is an accelerate subunit. So it's a heterooctamer together form the active complex, which is actually the shown here. A is the full complex containing 4 copies of PRMT5 in the middle, and then 4 copies of the MAP50 actually here. As you can see, at the current resolution 3.2 or 3.2 angstrom, you can clearly see the side chains of the bulky residues very well, and also the loop. There is a flexible loop right next to the active site somewhere here. As you can see, this flexible loop is actually quite flexible in the apple state. But once we resolved the compound bond form, the one in the middle, 11-2F, we can actually see that the flexible loop here, shown here, is actually much better ordered. Actually, this one. This is the flexible loop, which the flexible loop is ordered after the compound is bound to the active site. We are actively using this information to optimize this compound. The second project I'm going to tell you is a new method we developed for cryo-EM, which we called the chemically functionalized carbon films. The general idea is coming from the flash freezing step. As you probably remember, as I said in the previous slides, that during the flash freezing, we blot away majority of the specimen. Majority means about 99.9%. And then we leave a very small volume of the specimen across the grids and then freeze it. The idea is whether we can avoid this significant loss. So our general idea is that we can use a carbon film to cover a very thin carbon film to cover the Holy grid, and then make the inert carbon film active, which is actually the critical step in this technology. And then we can link different bioactive ligands onto the surface of the carbon film, and use this, we can selectively enrich biomolecular complexes we are interested in studying. And we have optimized this technology. We can put a large array of different ligands onto this surface. And people using similar technologies have actually optimized the modification of graphene oxide film. And there are multiple groups actually recently developed the technology based on our method, our idea. Let me show you, this works very well. Shown here is the first example is the nickel and Ta presenting carbon film. We can, using the chemical reactions to present nickel and Ta on the surface, which is still about 20 angstroms away from the carbon surface to give enough freedom. And then we can actually then quantify how many are on the surface. You can actually see, using the quantum dots, we can count individual groups that are able to bind to the his tag. We have a density about 1,000 functional groups per square micron on the surface of the carbon. And based on that, and based on our estimation, because of the entropy-related steps, our estimated affinity is about 35 nanometer. 35 nanomolar, which is about 200-fold higher than the affinity measured in solution. Exactly what we expected from thermodynamic calculation. Does it work on the grid? Yes. Shown here on the left side is our grid. On the right side is the conventional glow discharge grid. We load about 0.3 nanograms of a protein, which is about 600 kilodalton. It's very easy to see under the normal cryo-EM condition. As you can see, using our functionalized grid, we can see large number of molecules on the surface of the grid, but the conventional method, we see on very few. And if we do a calculation based on the surface density, we actually retained about 45% of the molecules we loaded onto the grid. As I said, we usually, using the conventional method, we lose about 99.9%. So we change from about 99.9% loss to about 50% loss. So we are about a few hundredfold better than the conventional method. The other typical grid you would use is actually RNA or DNA oligos loaded onto the surface. As you can see, we can, using our technology to do the same thing, we can put, say, this is an RNA grid. This is a DNA grid, actually. And we can put the DNA oligo on the surface and also do a similar counting, similar density, 600 to 700 functional groups per square micron. And then in the real grid, on the surface of real grid, you can also see on the left side is our grid, on the right side is our grid, but without the biotin, such that you will not be able to do selective attachment. This is just a negative control to see how many non-specific binding we experienced. As you can see, it's also a day-night difference. Our grid shows a dramatic enrichment of the loaded sample in low concentration. And this also can be used for three-dimensional reconstruction. Showing here is the example. We can put onto our RNA presenting grid, and then this is a 240 kilodalton protein on the RNA presenting grid. You don't have to know the details about the specimen. The general idea is that using this method, we can build a data set of the particles in different orientation and then perform three-dimensional reconstruction. This is actually just to show that even though the resolution is low, but the data is suitable for three-dimensional reconstruction. And this, in principle, can be optimized using a large number of data sets to reach atomic resolution. We are actually in the middle of doing that. Last is a recent development we are working on under the support of NIH. We can use this technology and load the transmembrane proteins onto the ChemC surface, and then add lipids into the solution, remove the detergent so that we can use the loaded membrane proteins to reconstitute a lipid bilayer on the surface. So here is some preliminary data. On the left is the surface with the protein. On the right is the same surface without the protein. As you can see, with the protein, we can actually form a bilayer. So the bilayer is doped with with biotin-related POPC, such that we can use streptavidin-coated quantum dots to directly label the big patches of the bilayer. You can actually see this. High density of the quantum dots means that we have the bilayer. Okay, so this line of development has been quite successful. As I mentioned, that multiple labs around the world are actually advancing our technology to different directions, especially using the graphene oxide. It's actually possible to further derivatize the graphene films and then do the same kind of presentation of bioactive ligands as we have shown. The third project is the project we did with James Chen's lab when my lab was still at UT Southwestern Medical Center. We used the technology to study the filament-based signal amplification in the innate immune response pathway. So a few words about innate immunity. So innate immunity means that the cells, a lot of the cells in our body has the capability to detect non-self molecular pathogens. For example, the S protein on the surface of the SARS-CoV-2, which is the spike protein on the surface. Though the peptides of the spike proteins are not actually presented are not present in our genome, which means that they are pathogen associated molecular patterns. So our cells have this capability to detect this pathogen related molecular signature. Proteins, peptides, DNAs, RNAs, or lipopolysaccharides and also other specific molecules associated with the pathogens can all be detected and trigger innate immune response. The one I'm going to discuss briefly is the recognition of viral RNAs. The same is true for the SARS-CoV-2, which has a positive sense RNA genome. The RNA viruses has this signature structure, which is we called the 5' triphosphate plus a very short double-stranded sequence in the RNA. With this signature can be detected either on the cell surface by toll-like receptors or in the endosomes or intracellularly if the virus are inside the cell and its genome are being replicated, its RNA signature can be detected by intracellular RNA sensor. This is called RIG-I-like receptors. There are multiple of them, and then these active RIG-I, so the RIG-I-like receptors or RLR, there are two main forms, RIG-I and MEVs, RIG-I and MDA5. I'm only going to discuss RIG-I, not MDA5. The RIG-I usually inside the cell is in the resting state and inactive. Once it recognizes the signature sequence in the viral RNA, it can convert into an active form. In a complicated sequence, it will then activate MEVs molecule on the surface of the mitochondrial membrane. And the MEVs change from the silent information into the active information. And MEVs has this N-terminal card sequence, card domain. The card domain is usually hidden in the inactive state, but in the active conformation, the card domain is accessible. So many of the card domains can come together and form this helical form a helical segment. Shown here is the data on the surface of the mitochondria. The MEVS is labeled with a flag tag on the N-terminus, and then after the viral infection, we using super resolution imaging, we can detect these short filaments on the surface of the of individual mitochondria. And this is the idea. The idea is that the released cut domain can mediate the filament formation, and then can expose the coil domain in the middle of the molecule. This coil domain, and this coil domain in the inactive state is not accessible, but in the activated state, in these filaments, they are accessible such that it will activate downstream signaling molecules. So we discovered, actually by EM, we first discovered that the filamentous form is actually the active form. So the collaborator, my collaborator working with us, actually developed an assay. So we actually first raised this point that maybe the monomeric form is not active, only the filamentous form is. So and because of that, we decided to study how this filament is actually formed from the individual cut domain. This is the cut domain. We can purify the individual cut domains and form the filaments. We can also purify the active domain of the full-length protein, except the transmembrane domain. This whole protein can also form a similar filament, except it has a lot of aggregates on the surface of the filament. We in this sample, we discovered that there were two different kinds of filaments. One type has C3 symmetry, the other has C1 symmetry. You don't have to know the technical details. They have different arrangement because of that. And we sorted them out. The C3 filaments, we can obtain the structure at around 9 to 10 angstroms resolution range. And then, and we also had electron tomographic data seems to apparently agreeing with the C3 symmetry. For the C1 symmetry, we were able to use the Titan couriers at Generic Farm and obtaining high resolution data up to actually the data quality is as good as probably like 2.9 angstrom. And using the computational algorithms, we were able to resolve the 3D structure at 4.2 angstroms. At this resolution, we are able to see bulky side chains pretty well. And using the X-ray structures of the cut domain, we were able to build the atomic model. And both the C3 and C1 model can explain a large number of mutagenesis data we have developed in the lab. I'm not going to get into details of the mutagenesis data. Shown here is just a few examples. Actually, you can see the residues involved in the molecular interface for building the helical filaments can be mutated and disrupt or destabilize the filaments, which will then dampen the signaling pathway from Rigi RNA viral RNA complex to MEVS. So as I mentioned, we have two different actually symmetry. This can also be demonstrated from the raw data. Actually, in the raw data, we can separate the C1 filaments and the C3 filaments, and then put them together. You can actually see the big significant feature in terms of crystalline packing or no helical packing is actually the relative strength of the layer line 5 and layer line 4. in the C1, as you can see, layer line 5 is almost invisible, but layer line 4 is clearly visible. In the C3 symmetry, you can actually see both layer line 5 and layer line 4 are clearly visible. Actually, we had a competitor at Harvard. They initially claimed that they don't have C3 in their data set. But if you look at their raw data indeed has C3 symmetry as well. And then, and the C1 the C1 projection map cannot explain the C3 feature very well. And then we did a careful analysis of all the data set we have on hand. We found that initially our data was significantly biased towards the C3 symmetry. That's why the C1 symmetry was not showing up very well in our initial analysis. Later on, when we started to make our specimen in low salt, we found that more than 50% or sometimes even 90% of the data is in C1 symmetry. So what's the real difference? The real difference is which one is really physiological or both are physiological. We still have no firm conclusion yet, but in literature, there is some data which is based on the C1 symmetry on the right side. This organization of the RIG-I match with the C1 filaments of MEVs, such that they would form highly stable complex, which would predict that in the active form of MEVs, or really in the virus infected cells, we should see clear co-localization of RIG-I and MEVs. But unfortunately, until now, we also tried significantly. My collaborator group has tried significantly. Also, many other labs have tried, both in cell imaging and also biochemically. We cannot detect the co-localization, either in cell imaging or in purified MEVS filaments. We could not detect RIG-I. But the C3 symmetry does not stabilize the complex between RIG-I and MEVS, which allow the dissociation of the RIG-I from the complex such that it can actually further amplify the signal. So I think currently the data seems to favor the C3 filaments is actually the physiological form because of the negative of co-localization data in both cell-based studies and biochemical studies. So recently, there is an adapter protein which was identified called Qiu-Xing. It was found that Qiu-Xing is actually very critical for the signaling pathway in virus-infected cells. So the general idea is that the RIG-I viral complex in the active state can bind to the N-terminal domain of Qiu-Xing, which can then actually activate the three limb domain in the C-terminal side. And then 3 domains in the C-terminal side can liberate 3 CAR domains from 3 inactive MEVs molecules on the surface of mitochondria. And this can then initiate the 3-stranded filaments to form on the surface of the mitochondria. This is actually, I think, favors the C3 filaments. So there is another unsettled point about the length of this double-stranded segment. X-ray crystallographers tend to think that this can be as short as 10 base pairs. But in our hands, in our assays, we found that 20 base pairs is actually required, which means that the minimum active form is actually a dimer. We suspect that after the monomer doctored to the 10 base pairs double-stranded RNA is actually activated, it will actually unwind the double strand. The unwinded zinc ion A can then allow the dimer formation, and then the dimers then becomes a radioactive form. So this will actually then make these observations consistent, but we still need further evidence to confirm that. So in the last, this part I demonstrate to you that Cryo-EM at different resolution range can really help to address very, very important biological questions in nature. The last part, I will tell you a story quickly about a major discovery we advanced and which we called lipid-dependent gating of voltage-gated potassium channel, or called KV channels. Voltage-gated potassium channels or voltage-gated ion channels in general are extremely important. Shown here on the left side is the action potential first recorded by K.S. Core in 1940s. The action potential has a rising phase, a decaying phase, and then a refractory period after that. In 1950s, an ionic analysis performed by Hodgkin and Huxley and their colleagues in England showed that the rising phase is contributed by the voltage-gated sodium channel, and then the decaying phase is contributed by the voltage-gated potassium channel. So they made two predictions. There must be voltage-gated and highly selective sodium and potassium channels. And second, these channels must have intrinsic gating charge that are responsible for sensing the change in transmembrane potential. In general, these channels work like this. You have the ion conduction pore in the middle, and you have the voltage sensing domain, which can sense the change in transmembrane potential. After the transmembrane changes, there is a conformational change in the voltage sensor, and this change is coupled to a conformational change in the pore domain, and switch of the pore domain between closed and open state. And in the open state, the pore can allow ions to go across the membrane. And there are two groups of channels: depolarization activated or hyperpolarization activated. You don't have to know the details. The general idea is that you can sense the change of the voltage of the transmembrane potential in both directions, either going to high or going to very low, going to negative side, can activate the channels. And they are very important. You know, they are voltage-gated potassium channels are in every live cell. They are needed to set up the resting membrane potential for every cell. And they are important for the proper function of heart, brain, and many other organs in our body, in the human body. And after the molecular cloning in 1980s and early 1990s, it was found that every voltage-gated ion channels must have this 4 helical bundles domain called voltage sensor domain, and every one of them contain 4 transmembrane segments, and the fourth one, which called S4, contains these regularly positioned positive charge residues, which are responsible for sensing the change in transmembrane potential. I'm not going to get into details, but the general idea is for almost all the known voltage-gated ion channels, either 4 of these subunits or domains come together to form one ion channel. Shown here is the example. Is an example of a KV channel. The middle is the pore domain, and 4 of them come together, and then 4 voltage sensor domains are flanking the middle pore domain. And there are many structures now available. I'm not going to get into details of the structures. What I'm just going to tell you is that it has been well worked out how the pore can achieve ion selectivity, and also how the voltage sensor domains can sense the change in transmembrane potential. One thing that's still not well understood is the exact movement of the body sensor domain, which we are going to work on. We are still working on. I'm going to tell you a little bit on this area as well. The direction I'm going to lead you to is actually this idea. The idea I started when I was finishing my postdoc training with Dr. Roderick McKinnon at the Rockefeller University. The idea is that because the body sensor domain is actually flanking the middle pore domain, is it possible that the body sensor domains are interacting with the phosphate groups in the lipid bilayer? We did a series of experiments in my own lab, and also many other experiments done by others are consistent with our main hypothesis. Our main hypothesis is that in a bilayer which does not contain phosphate groups in the head group region, the body sensor domains are favored in the resting state or resting conformation. In a bilayer which contains phosphate groups, the body sensor is favored in the active conformation. We have a lot of biophysical evidence supporting that. The question is really whether there's any physiological relevance here. Actually, if you look at the native lipids, there are two groups. Group 1 is what we call the phospholipids. Group 2 are what we call the non-phospholipids. The very famous example is a well-known one, cholesterol. Actually, in some of the bacteria, they are cholesterol analogs. And actually, around this, 40 to 50% of the lipid molecules are non-phospholipids in the human cells. In some of the cells, this can be even higher. For example, red blood cells. We already demonstrated that the lipid-dependent gating has significant effect in the in vitro system. People in the past 20 or so, 30 or so years has accumulated enough evidence that in the isolated cells or in cultured cells, there are significant effect by loading cholesterol, enriches the cholesterol content. It can incur significant inhibitory effect consistent with what I discussed, what the lipid-dependent gating. The real question is whether there is in vivo effect. So for this, we are currently working on with knockout mice or knock-in mice, which can change the cholesterol content in the cell membrane. But if we generally look at the genetic mutant in human patients, what we find out that so far almost all the lipid metabolic defects in human patients affecting the cellular content of non-phospholipids. Our prediction is all these patients must suffer significant neurological defects because of the effect on the voltage-gated ion channels in the neurons in the central nervous system. Actually, there's no exception. All of these patients suffer severe defects since infant period. So we are working on three conformational states: the resting state, the open state, and the inactivated state for these KV channels. People have been working on 2D crystals, small vesicles. We are not working on. We are currently using biocells, detergent myocells, and nanodiscs to study them. So we have three different conditions. We can show here are the Cryo-EM images. I will quickly go through. I'm running a little behind. Shown here is the one which we found a condition that it can keep the channel in the resting state. We are currently at around 8 angstroms. Under this resolution, we can actually recognize the individual alpha helices, actually, even though we cannot reliably designate them. But with the structural information we know about this channel, we were able to build a tentative model, this is actually the body sensor part, S4 and S3. As we can see from this tentative model, the S4 is actually moved vertically about 10 to 15 angstroms, such that you will actually switch from facing outside to facing intracellular side. For the inactivated state, we build we have a nanodisk bound. We have the channel in nanodisk, which is bound to the FV, an immunoglobulin domain, which allow us to actually align the particles. We have been able to get a nice data set reaching 3.6 angstrom resolution, which allow us to actually build a molecular model in this conformation. In this conformation, we can actually see that the vortex sensor is actually undergoes a significant rotation, no movement in the lipid bilayer. Instead of vertical movement, it's actually rotational movement in the bilayer. And then because of this change, the change in the vortex sensor, the S6, this is the intracellular half of the S6, is relaxed such that it will actually swing from the intracellular membrane from the middle part actually downward such that it will actually close the pore. Using the atomic model, we identify the residues that are actually swing towards the middle of the pore and then keep the pore closed. This is how the steady state inactivation happens. This is actually a conformational state that has been looked for by many people in the field. We are the first one. So using this conformation, we were able to show that the vortex sensor seemed to move vertically for the resting state and horizontally for the inactivated state. With all this, I'm ending this talk. So I have showed you that Cryo-EM is very useful, very versatile tool, deliver the structures in either intermediate resolution range or at near atomic resolution state. And both resolution range, in both resolution range, the structural information can be used for revealing important biological insights. And using the near atomic resolution structures, it's actually feasible to perform structure-based drug design. Lastly, the slide shows the people currently in my lab at Hauptman-Woodward Medical Institute and former members with me at UF and UT Southwestern, and my collaborators in different institutions and funding agencies paying the bills. Thank you for your attention. Hello. I'm done. I'm running a little behind. I don't know if you have, we still have time for a few questions.
Hi, Jiang. This is Silke from Silke Yevdokimov and Artem Yevdokimov. Can you hear me?
Yes, please.
I have many questions because I really don't know a lot about this, but I loved it. Thank you very much for sharing all those information, and your work has been great. Maybe so now get to meet you in person and
Yes.
you can teach me all this. I loved it. Thank you.
Thank you. Hello, any questions? You can also email me with your question later.
No, looks like no question.
Yes.
No.
Thank you, Qiu-Xing. That was a great talk.
Thank you.
Hello.
Yes.
I had a small question about your project 3, where you said that based on the observations, you concluded that it might be C3, that is the physiologically relevant form of this filament.
Yes.
And I remember you showing data that said that C3 usually forms when the conditions are high salt.
Yes, high salt is actually around 150 to 200 millimolar, which is actually physiological.
That is exactly I wanted to know, and how relevant it is when you compare it to the biological salt conditions. Thank you very
So
much.
the inside the cell, which is about about 140 to 100 to 150 millimolar potassium chloride, and there are also 2 to 3 millimolar magnesium chloride, very low concentration of calcium, of course. There, so it's very close to the high salt condition we used.
Yeah,
The low salt condition we used is usually below 50 millimolar.
Thank you. I just wanted to know that.
Thank you.
Hi, Jiang. I have
Yes.
one question.
Yes.
It's regarding the nickel NTA coated grid that
Yes.
using the new method.
Yes.
So in that case, all your molecules will be in like kind of similar orientation.
No, it's not. So yeah, thank you. This is a very good question, because we actually worried about this as well. It's actually not. I mentioned during the talk that the chemical groups, the nickel NTA, is about 20 angstroms away from the surface, such that, and also the linker between is actually quite flexible, such that the orientational change of the molecules are still kind of fairly random. We actually then used the C3PO as a complex to demonstrate this point, that using this grid would not restrain the freedom of orientation for individual molecules. Mm-hmm. And then use the images, we can build a three-dimensional map without any problem. It turns out that if we compare it with the samples stick to the surface of the carbon, our method is actually better because of the spacing effect.
Mm-hmm.
So we don't have that problem.
Mm-hmm. Thank you.
Okay, and also we're going to make the slides available through our institutional website. So, and you're welcome to look at it later. And then I will hopefully put the, I think my email address is going to be online as well, so it should be fine. Any more questions? Okay, without any other question, I guess we're going to close this talk. Thank you for your participation. I look forward to meeting with you all in the future. Thanks. Bye.